So hello everyone, and welcome to another conversation of the fourth cycle of conversations on corporeal architecture. My name is Maria da Piedade Ferreira, and today I am very glad and honored to welcome Kate Jeffery. Kate Jeffery is a medically qualified neuroscientist. Her lab studies rats to find out how sensory information is assembled by the brain into more complex cognitive representations of the world. It focuses on the cognitive map, which is formed by the hippocampus and associated structures, and underlines our sense of direction and sense of place. She is also interested in the link between architecture and the representation of space in the brain, and is researching this using virtual reality. Kate, thank you very much for accepting this invitation and being here with us today.
Thank you very much for the invitation. I'm just going to share my screen and start the slideshow. Can you see that?
Yes, we can see, but we also see the next slide.
Yeah, okay, so you see that that's because I've got two screens. Let me try this one. Is that better?
Yes.
Okay, great. Yes, so I just want to talk about the work that's been unfolding in my field in the last few decades, really. Thinking about space and how the human brain constructs a kind of an internal representation of space. And the way that we've been coming at it from the point of view of neuroscience has been slightly different from architecture, because architects have tended to focus on the emotional components of space, so how spaces make us feel. Whereas in psychology and neuroscience, we've been exploring what we call cognition, knowledge, so how the brain understands space. And I think, I've come to the conclusion over the years that this is actually a critical part of how we feel about space, the fact that we need to be able to understand it in order to be able to make a mental representation of it. And so what I want to do is to tell you a little bit about what we've learned about how the brain constructs its knowledge of space. So the brain is very complex, but when you look at it, it's very simple, and it's kind of hard to imagine how all of our thoughts and feelings and knowledge and so on can be built by something like this. And really the way that, oh gosh, so this is automatically, let me just kill the slideshow. Sorry, it's got the automatic slideshow timings on it. Let me try this again. Okay, we'll get there. Can you see that?
Yes.
Okay. You can see the whole screen?
Yes, yes.
Okay. So yeah, Kevin Lynch, who is an architect, started thinking from an architect's perspective about how it is that we construct knowledge about space. And he identified these elemental aspects of space that to his mind were important for understanding, or what he called imageability of a space. So he was thinking in city scale terms. How do we image the city, make a mental representation of it? And he identified that there were these core elements: paths, edges, districts, nodes, and landmarks, and that the brain is having to kind of learn about these things and understand their relationships. Funnily enough, we as neuroscientists have converged on more or less the same view, actually, when we've looked inside the brain and tried to see how the brain is building this map of space. We find that we see the same elements. We see paths, edges, districts, nodes, and landmarks. And these we've found by studying rats and mice mainly, but we also know that humans have the same mental machinery. So It's something that evolved very, very long time ago, probably a long time before even the common ancestor of rodents and humans. So maybe hundreds of millions of years ago. So these are very fundamental processes that we're looking at. So in thinking about designing space, we're dealing with these kind of building blocks. So how do we build more imageable cities is one question. And to understand how to do that, we need to kind of know what's going on inside the brain. And I mentioned the emotional components of space, and there's a lot of evidence that an inability to image a space has very negative emotional consequences. For example, when people are unable to comprehend a space, they become very anxious, and this is a kind of a feeling that I've termed spatial unease. And a really good example of this is the Seattle Central Library, which has won all sorts of awards. Architects love this building. And it is beautiful to look at. But the first day that it opened, people were complaining already that they were getting so lost that the staff were needing to put signs up with stickers everywhere to help people find their way around. They couldn't find the books, for example. So it was a beautiful building aesthetically, but cognitively, it was very difficult to understand. So it wasn't imageable. So how does the brain build an image of a city inside itself? So here's just a diagram just to show you the elements of the brain. So the brain is quite modular in some ways, but not completely. So there are different parts of the brain specialized for different functions. For example, the bit in green is specialized for primary vision, so recognizing the simple components of the visual scene. And all of the senses have their own primary areas that are the first parts of the brain to receive the information that's coming in from the sense organs. So we've got primary vision, we've got primary audition, taste, touch, all of these senses, then feed that information to what's called the secondary association areas. So these are the parts of the brain that combine all this information and start to build up our kind of understanding of the world, really. So that's the cortex, which is the outer part of the brain. If we look inside the brain, we also see that there are structures underneath that are receiving information coming in from the cortex and then doing things with it. And one of the structures that's turned out to be incredibly important for space and also for memory is this structure called the hippocampus. Okay, so if we peel away the cortex, we see these subcortical regions. These are the bits underneath. Some people think of this as kind of the ancient brain. And one of the subcortical structures that's been incredibly important in our understanding of space is the hippocampus, which is this structure here, which is very long and thin in humans, but actually very short and fat in rats. And it was discovered quite a long time ago that damage to this structure produces two things. It produces an amnesia, so a loss of memory, and it also produces a spatial confusion and unawareness, so people become unable to navigate around space. And so that was the first clue that space and memory are very, very intertwined in the brain. So a lot of these structures, these subcortical structures, are also involved in things like emotions, and so they're very strong interconnections between the emotional systems in the brain and the spatial system, which makes sense because we do have strong emotional attachments to space. So one thing that we've learned by studying the neurobiology of space is that there are more than one kind of spatial system in the brain. So we can actually make a distinction between local space, which is reachable space that's in the region surrounding your body. So, things that you can reach with your limbs. This is actually controlled by a different part of the brain than the part that shapes your awareness of large scale space. So space that you can walk through and navigate through. So it's possible to have a dissociation between these things. For example, to have a stroke that affects your ability to understand where things are in relation to your body, but not to find your way around the house. Or conversely, you might have some type of damage to the hippocampus, that means that you can no longer find your way around the house, but you can still pick up a coffee cup. So there's local space, and then there's navigable space, you could call it. So the local space is controlled by this region called the parietal lobe, which is one of these cortical regions that I was telling you about. And the more global space is controlled by the hippocampus. So this is a really nice example of what happens if you lose your local space, which is important. You know, when you're trying to build up a global map, the first thing that you do is look at where things are in relation to you, if you're in a space that you don't already know. And people who have had a stroke that affects their ability to process local space often have these strange symptoms where they simply don't understand what they see in front of them. So for example, somebody who's had a stroke affecting the parietal lobe may be unable to draw a clock. So they can draw the right-hand side of the clock, but not the left-hand side of the clock. Or they may eat all the food on the right-hand side of the plate and not the left-hand side of the plate, shave the right side of the face and not the left, and so on. So our kind of understanding of left and right is controlled by the parietal cortex. On the other hand, our understanding of how to find our way around our house or around our city is controlled by the hippocampus. And we also know now by the regions of the brain outside the hippocampus. There is also a third system in the brain which is responsible for guiding actions. And this is called the striatum. And this is part of the brain that is what we think of as kind of controlling automatic or habit-based actions. So when you're navigating on autopilot and not really thinking because you know the route really well, for example, you know, you may be able to get yourself to your office without even realizing that you've done it because you were lost in thought. And that's because the striatum is able to take the spatial movements that it's learned about over and over and over again, and then just execute those without your conscious mind having to think about it. So the striatum comes into play when we've got actions in the world that are so well learned that they're very predictable and reliable. And so, you know, the brain has decided it doesn't need to think about them anymore. So it will make those automatic. So when you're kind of navigating around a very familiar space, you may be using your striatum. Striatum is quite deep in the brain. It's shown in red in this picture here. So this is an example of two situations, one of which you might need the hippocampus and one of which you might need the striatum. So on the left is your commuting route to your office. It's very well known. You're doing it on autopilot. If your automatic behavior becomes not helpful for some reason, for example, you encounter a roadblock and your automatic route no longer applies, then your conscious brain is called into play, and it probably involves the prefrontal cortex, which is the kind of thinking and planning part of the brain. And it will switch on the hippocampus and say, you need to use your map to figure out a new route to the office. So these multiple different regions of the brain are all kind of interacting and shaping our knowledge. So how's all this done in the brain? So we've now got some amazing techniques for looking into the brain and seeing what's going on inside. And one of the most helpful for studying humans has been functional neuroimaging, which allows us to see what parts of the brain become active when somebody is thinking in a way that's using the different parts of the brain. So for example, this is a brain scan of somebody who was doing some type of navigation task. So they'll be doing it in a scanner. You can see the scanner in this photo here, and probably with a computer screen in front of them, they'll be watching a video or something like that. So this is an area of the cortex called retrosplenial cortex, which I've become very interested in. And I've been studying in rats because it seems to be involved in helping the brain use landmarks to be able to work out where it is or where you are. So the visual system sees the landmarks, and so that primary visual cortex that I mentioned passes it to the retrosplenial cortex. And the retrosplenial cortex is talking to the hippocampus and saying, this is what we can see. And the hippocampus is saying, well, this is where I think we are. And those two things get linked so that the next time you go back to that place and you see those landmarks, you will know where you are. So it's very, very important dialogue between these two brain structures. Another technique that's been really useful is virtual reality. So we can now create environments that we can manipulate in subtle ways. For example, in a brain scanner, obviously you can't walk around a real room in a brain scanner, but you can walk around a virtual room. And we've also got virtual reality for rats and mice. So this is one here. This is an example of a mouse. What the mouse does is it runs on a little ball. And as it runs, a computer detects the ball rotating, and it makes the video move past the eyes of the mouse. So from the mouse's perspective, it's running and the world's moving past it. And we think that that simulates real movement through real space enough that we can then look at the neurons and see what they're doing as the mouse is kind of moving around this virtual world. It's all pretty amazing technology. So brain scanning in humans lets us broadly see which areas of the brain are doing what. But to really understand the language that the neurons are using, we need to go right inside the brain and to pick up the action potentials, the nerve impulses that the neurons are making as they're doing what they do. So it's like listening to a language and trying to decode it. So this is a cross-section through the hippocampus. This was of a mouse, and the mouse had been genetically altered so that its neurons, as well as doing all of the normal things that they do, they also make a protein that shows up with a color if you shine a fluorescent light on it. So these neurons are just making this protein. And it means that when fluorescent light is shone on those neurons, you can see the many different neurons that go to make up the hippocampus. It's really remarkable. And you can also see the processes, the axons, coming out of the neurons and connecting them to each other. So you can now start to trace pathways through the brain. And what this is a schematic of an electrode, which is a tiny little wire, the thinness of a hair, that's been inserted under anesthesia into the hippocampus. So that means that we now have an electrical pathway between the neurons and the computer, if you like. So as the mouse is kind of running around doing whatever it's doing, we can actually listen to the neurons talking to each other. So we have used rats and mice partly because we can do these types of invasive experiments in rats and mice that are very difficult to do in humans, although actually not impossible. So we use that electrode implantation technique in humans with epilepsy to try and monitor where the focus is prior to surgery. And so it's also been possible to record from some humans. And we see the same things in humans in virtual reality as we do in the rats and mice in real reality. So we're pretty sure that we have the same brains, even though they're much bigger. So this is a schematic showing you the hippocampus. And as I mentioned, in humans, the hippocampus is very long and thin. And this is it way down in the temporal lobe, which is the parts of the brain sort of down here behind the ears. In the rat, the hippocampus is very much larger. And this has meant that when we put electrodes down from above, the first thing that they encounter is the hippocampus. So it's been relatively easy to study what's going on in the hippocampus. So in the 1970s, my old boss, John O'Keefe, made this really amazing discovery by recording in the hippocampus, which is that if you have a rat running around on the surface of an environment, and you have one of these wire electrodes in the hippocampus and you're feeding the signals into a computer, what he discovered was that those, the action potentials produced by the neuron, which are these spiky things here, the action potentials emitted by a single neuron, a particular neuron, will all tend to occur in one place in the environment. So here's a schematic. So if you look at these 4 action potentials and you mark them with a little red square and then plot them on where the rat was at the moment that those action potentials were emitted by the neuron, so these are little electrical pulses, you can see that that neuron was only really active when the rat was in the southeast corner of that platform. So it was walking around all over the place. That's what the squiggly line is. But every time it goes into the southeast corner of the box, suddenly that neuron becomes really active and fires lots of action potentials. And this was kind of a very unexpected discovery because, for one thing, nobody had really linked the hippocampus to space at that point. But also, when people had recorded neurons prior to that time, the neurons had tended to kind of respond to very simple sensory information, like visual information going across the retina or something like that. This idea that this neuron is active, even though the sensory information coming in is really variable, because sometimes the rat will be running in this direction and it'll be able to see some part of the room, and when it's running in the other direction, it can see a completely different part of the room. But the animal might close its eyes briefly, that you can turn the lights out, you can really change the sensory information, and yet the neuron always fires in that place. And so O'Keefe proposed that these neurons are what he called place cells. So this is just an example of two place cells. Again, it's a schematic. We're now looking down from above, and you can see that one of these cells is producing all of its action potentials in the northwest corner of the box, so that's the red cell, and the other one is producing all of its action potentials in the southeast. And there are hundreds of thousands of these cells in the hippocampus. So between them, they can completely cover the entirety of the environment, and indeed the entirety of any environment. So if you were to take that rat out of that place and put it in a different place, the cells would still be active, but they would all be active in a different way. There'll be different cells active in different places. So there's a new map for every environment, and there's always a map. So the brain is always making maps. It was then found that the damage to the hippocampus causes rats to be lost. I'm not going to go into this just for time reasons, but there's a sort of a test for rat navigation called the water maze test. And rats with damage to the hippocampus are unable to find their way around in this tank of water. And it was also been well established now that humans with damage to the hippocampus can't find their way around. And also, if you put them in a brain scanner, so Eleanor Maguire was the first one to really show this convincingly. If you put them in a brain scanner while they're navigating, then the hippocampus becomes active when they're planning where they want to go. So she's really famous. She and Hugo Spears showed that they studied London taxi drivers because these people have a very, very detailed map of the entirety of London in their brains. And so they got them to imagine having to drive their taxi from one place in London to another, and found that sure enough that the hippocampus became active. And also that part of the hippocampus, the spatial end of it, became larger, or at least was found to be larger, in the people who had been taxi drivers for longer. So it sort of starts to seem like maybe using the hippocampus a lot may cause physical changes in its structure, which was pretty amazing. And then, as I mentioned, we've been able to record these cells in people with epilepsy as well. And we also find when they navigate a virtual taxi around the virtual environment, we see the hotspots of activity in the neurons. So the neurons become more active when they're navigating this virtual taxi around this virtual town. So it's all kind of adding up to the hippocampus is important for space. And not just for real space, but also for imagined space. So shortly after place cells were discovered, or at least it wasn't short, it was about 20 years later, short in science terms. It was found, so people started looking at the parts of the brain near the hippocampus that are sending information in, and they found that there are cells in the neighboring regions of the brain that seem to be also doing spatial things, but different kinds of spatial things. So for example, it was found that neurons in this region here are active all over the place. So they're not like place cells that are only active in one part of the place, but they're only active if the animal faces in a particular direction. So every time it faces towards the door, let's say, this particular cell will become active. And then when it turns its head, that cell will stop and a new one will start. And so there are all of these different cells for different facing directions, but they're all active everywhere. So they don't care where the rat is, they just care which way it's facing. And they called these cells head direction cells for obvious reasons. And they proposed that these cells are acting a bit like a compass, sort of telling the hippocampus which way around the animal is facing, so that it can then kind of figure out where it is and which way it's going and so on. So these head direction cells have been found in quite a few brain regions now, just a few of them are shown here, but it's been found in many places. So a lot of the brain is interested in which way we're facing. And this to me is one of the most important findings that's come out of the study of the neurobiology of space. because it's something that I don't really ever hear architects talking about very much, the sense of direction and how important it is for our understanding of space. So if you don't have a sense of direction, then the world is just really confusing. Even if you can see perfectly well and you can see where everything is, if you don't have a sense of direction, it seems to be very difficult to make a mental map. And you can show this by messing around with your sense of direction. For example, if you put on reversing goggles that switch left and right so that your sense of direction, doesn't match what you can see, that becomes extremely confusing. So sense of direction is absolutely fundamental. And I think that one of the biggest things that architects could learn from neuroscientists is to think about this when designing for people to navigate. When you're creating a building, what is the information that the head direction system can use to orient itself? And how do you provide that information from the moment the person sort of sets foot in that space? Because the head direction system very quickly needs to know which way around to organize itself. And if you don't give it the right information, it'll just take a guess. And if it guesses wrongly, then you'll be all messed up. So the head direction cells are really important. And then finally, well, not really finally, but another really important discovery that was made about 15 years after the head direction cells is these cells in the brain, which were called grid cells. And I'm not sure if this is going to turn out to have been the best name for them in retrospect, but these grid cells are like place cells in as much as the actual potentials produced by the cell are all emitted in not everywhere in the environment, but only some places in the environment. But for grid cells, it's multiple places. So whereas with a place cell, the cell will only be active in one place, a grid cell will be active in lots of places. And the really interesting thing about that is that the places where it's active are evenly arranged, evenly spaced in these kind of neat rows where there's the same distance between each place. So for example, the cell is firing here, and it's firing over here, and it's firing over here. And the distance between those places is the same. It's around about, for the cell, it's around about 30 centimeters or so. So it's made this really incredibly regular grid-like pattern, which forms a pattern that we call hexagonal close packed. It's a pattern that's well known to mathematicians and physicists. So it's kind of hexagonal because for each element, it has 6 nearest neighbors. That's true for every element. So this one, if the environment was a bit bigger and the rat would have walked a bit further, it would have had 6 nearest neighbors as well. So, so each, what we call firing field, the place where the cell fires, each firing field has 6 firing fields around it. So it makes this really beautiful hexagonal pattern. And so our immediate thought when grid cells were discovered is, are these literally a grid for the brain's map of space? And it's made me sort of do some wondering about whether there's something special about hexagonal symmetry for our brains. And we, maybe we have a predisposition to like hexagonal symmetry, or conversely to dislike hexagonal symmetry, or maybe it's not relevant. We don't know, but sort of interesting food for speculation. So these discoveries were considered very important, and O'Keefe got half of the Nobel Prize in 2014, and the discoverers of grid cells, the Moses, got the other half. I think head direction cells should have also gotten a look in, but Nobel Prize is very restrictive. So. But that's just an indication of how important it was considered as a discovery, because it's really opened up our understanding, not just of space, but also of how the brain is organizing knowledge. So we've discovered lots and lots of different cells now, but these are kind of what we think of as the canonical spatial cells. I didn't mention border cells so much, but border cells are also found in the same place as the grid cells, and in the part of the brain that sends a lot of projections to hippocampus. And we think it's, so these cells become active when the rat is against one of the borders. and different cells like different borders. So clearly there's some type of directional information telling them which border is which. And we think that this might be important for helping to kind of anchor the spatial map. So going back to Lynch, it's sort of like we're having the ingredients of the spatial map here. We've got the borders, we've got the landmarks, which I haven't talked much about these, but this is how the head direction cells know which way around they should be. We've got the district, which is possibly represented by the place cells. Path, we have paths, we have evidence of path encoding in the brain too, because the place cells, when a rat walks along a path, the place cells become active in a sequence as it's walking. So first cell will fire, and then the next one, and then the next one. And that sequence recurs at later times. So if the rat then sits and stops and thinks, or maybe has a bit of a nap, when we record the place cells, we see the same sequence appearing spontaneously in its brain while it's dreaming or thinking or whatever, as that same sequence that it walked through when it was awake. So we think that what's happened is the brain has learned that sequence and is now practicing it. So it's rehearsing it so that it can store a memory of that path. So these elements of a spatial map are all present in the brain. So just to cover a couple of things that we've learned, sort of take home messages, I guess, from all of this. So we immediately, as soon as place cells were discovered, it was evident that the boundaries of the environment are important, because you can move, the rat is in a box, or it's on a platform, or whatever, it's in some bounded space. You can move that space in the room, and the place fields will move as well. So clearly they are using the boundaries to create the reference frame within which they establish their firing fields. So then you can sort of play some tricks on the cells and say, okay, well, what happens if instead of moving the box, we expand it? And what we didn't know what would happen a priori, because you could imagine that maybe what the cells will do is that some of them will follow one of the walls, and some of them will follow the other wall, or maybe everything will just kind of stretch out. And actually, so this slide is very slightly misleading, but some of the cells did stretch out their firing fields. In other words, the range of space over which they would be active stretched. Interestingly, it didn't stretch as much as the box had stretched. So if you increase the box dimension by 50%, then the place fields would typically only stretch by half that, by 25%. But some cells didn't stretch at all, and they tended to be the ones that were right near the walls. So it sort of looks like the closer to a wall a place cell likes to fire, the more it's going to use that wall in preference to anything else. Whereas if it's a cell that likes to fire in the middle of the space, then it's probably using more than one wall. So that's kind of got us thinking about how the brain is processing the space that you're in, and the boundaries, and how far away they are. And if you play a trick on someone and stretch the room, and indeed they've done this in experiments, what happens to people's perception of where they are, and how do they reconcile the conflicts? And so there's lots of fun to be had with trying to fool these cells and mess around with people's sense of space. So interestingly, the grid cells also stretch, and again by only 50% of the actual stretch. So the fact that it's only 50% is telling us that the cells are trying to find a compromise between two sources of information. One is the boundaries, but the other one is the intrinsic sense of motion. So as the rat is walking, the brain is processing how far it's walked. It's got an internal odometer or speedometer or whatever. And it sort of knows when the rat has walked around about 30 centimeters. And that's how the grid cell knows how to position its activity at 30 centimeter intervals. So you've got the internal signal that says this is 30 centimeters. And then you've got the box, which the rat's just learned about, which says, these walls are supposed to be a meter apart. And once you've stretched the box, there's a conflict between those things. And the grid cells resolve this conflict by compromising where they fire. We don't yet know, again, quite what that means for the rat or the person's internal conception of space, but it's kind of interesting. The other thing is you can mess around with the symmetry of the pattern by just putting animals into an asymmetric environment. And we're still not sure what that means. But it's interesting, and it reinforces the importance of the boundaries for the cells trying to make their map of space. So again, this is something I'm not sure that architects, I'm not sure how much emphasis architects place on boundaries of an environment. But I think it's becoming apparent that these are really important for our map of space, along with direction. They're so important that they can override this intrinsic, footstep counting mechanism, because you can create an environment that has repeating elements, and then see what the cells are doing. So from the point of view of the rat, you would think that as it's walking in and out of these different rooms, then the rat knows that it's walked in and out of different rooms. It's kind of knows that it's walked along a corridor. So in theory, it has information that these rooms are not the same. But when the rat goes into one of those rooms, by and large, the place cells and the grid cells will do the same thing in every room, as if the rat is just going in and out of the same room. So it looks like the local characteristics of the environment, the boundaries and what the rat can see, dominate over the other information. So we don't know whether the rat really does think it's just going in and out of the same room, or whether the rat is smarter than it cells. But we do know that it's easy to confuse the place in the grid cells with these repeating elements. And again, this is something that architects probably already know from humans. And it's something that potentially you can have some fun with if you want to confuse people. You give them multiple repeating rooms. Interestingly, the confusion is worst if the rooms all have the same orientation. But if the rooms have a different orientation, the brain is very good at knowing which room is which. It's much less confused. And that's because the head direction cells are able to sort out the confusion and say, I know these are different rooms because I know that north is towards the door in this room and it's towards the corner in this room, so there must be different rooms. So the head direction system, provided that it's not also confused, is able to disambiguate spaces that look the same, provided they are oriented differently. And I've mentioned how important direction is, so I'll just skip over that. The final thing to say is that you can have rooms that spatially are the same, but there's something different about them. And maybe they look different, or maybe even they look exactly the same, but something different is going to happen in them. So for example, you could have a room that during the day is a sports hall and during the evening is a concert hall or a theatre, same room. But it's being used for a different purpose, and people go in there with different expectations. And that type of internal knowledge is enough for the place cells to make a new map. So I think that's very interesting, because it sort of suggests that really what the place cells are all about is not just space in isolation, but really space plus meaning, or what we call context. And in fact, I've wondered if we ought to be calling them situation cells. So their job is to identify these places in space where certain conditions pertain, like this is the sports hall at the moment, and then to create a map specifically for that situation. So the final thing I just want to leave you with is that, so some of the work that I've been doing in my lab has been looking at what happens to the space, to the place cell and grid cell maps if the environment has complex structure. And this is just a synopsis of various experiments that have been done, some in my lab and some in other labs, just to say that the place cell pattern can be different depending on the structure of the environment. So for example, if rats are running backwards and forwards on a linear track, then place cells will make a different map going one way than going the other way. So the hippocampus has decided this is two different situations, running this way and running that way. And yet that's not true on an open field where the rat can walk freely in all of the directions. Now those cells don't distinguish running in one direction from running the other, because the system has decided that's a single situation. I'm in this space. So that's an example of how the hippocampus is trying to parcel up things into situations that are meaningful. We've also looked at 3 dimensions, what happens if the animals can go vertically. And we're sort of finding some mixed mixed results depending on how the animals are exploring vertically. So for example, if the animal walks around and around and around on a spiral staircase, we get place cells that are active on multiple contiguous coils, as if that cell is trying to represent this region of three-dimensional space. And so each time the rat goes around a coil that cuts through that region, that cell will be active. So it's multiple coils, but it's not all of the coils. And when it's multiple coils, they're always contiguous. So that sort of suggests that maybe there's a three-dimensional kind of place cell map. And actually, when we've looked at a truly three-dimensional space, a lattice where the rats can climb through, we do see that places where the place cells are active form these three-dimensional shapes. So it looks like there's a three-dimensional place cell map space, which is kind of cool. With grid cells, it's a bit more complicated. We see, again, that they make a different pattern going one direction than the other. So the cells are treating this as two different situations. They make this nice pattern that we've already looked at on a two-dimensional surface. When the animals can go into three dimensions, on the spiral staircase, it now looks like the grid cell is different from the place cell, because the grid cell does the same thing on every coil. It's as if that cell doesn't know how high the rat is and doesn't know about multiple coils. It just thinks the rat's going round and round and round the same coil. So that led us to think maybe the grid cells tracking distance that the rat has walked are not sensitive to vertical distance. So then we've looked at what happens if the rat can actually walk around on a vertical wall. And now we see that the grid cells are sensitive to vertical distances, but the scale is really different. So they fire over much bigger areas and the patches are much further apart. It's as if the whole map is vastly expanded. And we have no idea what this means. It's quite intriguing. Does the rat think that the space is just much smaller than it really is, and so it has to walk much, much further to cover 30 centimeters? We don't really know. So we haven't yet done the behavioral experiments to ask the rat, but it's very, very intriguing. And then finally, in three dimensions, we do see the blobbiness of, the same type of blobbiness that we see in two dimensions from the grid cell pattern, except these are now spheres instead of flat circles. But it's not regular in this kind of nice, neat hexagonal pattern that we've seen on a two-dimensional surface. I'm trying to understand what does that mean for the brain's conception of space, and still trying to work that through. Does that mean that the rats don't have such a good conception of where they are in three-dimensional space? Or does it mean that the brain doesn't actually care about the regularity of that grid pattern? It just cares about the spatial separation of those spaces. So we don't yet understand. There's a lot of work to be done to try and untangle all of this. But really, I suppose the take-home message from this is that the map is not a map of a space out there in the world. It's more of a representation of the things that you can do in that space. So it's a map that's very much tied to the actions that you can make. So boundaries, for example, that limit where you can go, whether it's vertical as well as horizontal and all of this kind of stuff. So we're developing a very action-oriented view of how the brain is conceptualizing space. And I think that's going to be really important for architecture. So people may perceive a space really differently, depending on the affordances of that space for different kinds of movement. So for example, somebody in a wheelchair may perceive a space really differently from somebody who can walk around. So I think this action-oriented view is also emerging in other branches of psychology, and it's quite interesting to think about. Okay, so I'm going to stop there. So I've talked for quite a long time. I don't know how to stop sharing. So
it's fine, I can do it for you.
Thank you.
Okay, so can you see me and hear me?
Yes, I can see you, yeah.
Great. So thank you so much, Kate, for this really interesting presentation. We were here in the room very focused, and I was also very focused on thinking also about the first question to kick off. So I would first ask you if you could perhaps tell us a little bit more. Now your last slides already went into this direction. When you're designing your experiments, in some ways you're also working as an architect, because you are designing the spaces where your rats are going to navigate. So what kind of design parameters do you mostly work with when you're designing these environments? So you had this example, for example, of the spiral staircase, and we saw also a few ramps, but do you also work, besides everything you know about place cells and all of these different elements of the rat's brain, do you also design this environment thinking about their motivation, how to trigger them to allure them to navigate a specific way? Could you perhaps tell us a little bit about it?
Yes, yeah. So we do need to use motivation to get them to explore. Not so much with mice. Mice tend to be really active anyway, but rats, they're quite lazy and they'll just curl up in a corner and snooze unless you give them a reason to explore. So what we normally do is spread food evenly around the space to encourage them. So they can't really detect it. So that's like tiny bits of rice and so on. They can't really detect it. They have to hunt for it. So that keeps them really busy exploring, walking all around. So to be able to record these neurons and to build up the spatial pattern, it's really necessary for the animals to have homogeneously covered the environment. But that's a little bit unnatural for rats. Like a normal rat wouldn't do that. You know, normally they come out, they go straight to where they know there's food, they get their food, and then they scurry back to their nest because otherwise a hawk will get them or something like that. So what we're seeing, so we're recording in a situation that's not entirely natural. And I have wondered whether the really nice symmetrical pattern that we see with the grid cells is an unnatural manifestation of that unnaturally homogeneous behavior, and that really a normal rat wouldn't normally have grid cells that produce a nice hexagonal pattern like that. Mm-hmm. So yeah, the pattern of exploration is important.
This is also very interesting to understand. And did you ever notice, for example, in the experiments that even with these little motivation enhancers, the food and so on, did you notice if the rats had any actions that show that they would like to transform the space they're in, as if they would have perceived, for example, if they would have bumped into a limit or a wall that perhaps trying to open it or create an opening by crushing it? Did you notice any tendency to transform the space creatively?
I'm not sure that I would say creatively. I mean, certainly they will look for openings, and because they like to not be confined in a space. So if you put a doorway in the wall, for example, they'll figure out how to open that door so that they can go through it. But that's not really, I think that's not really what you mean. I think just in terms of do they build their own spaces? Is that what you mean?
No, really being in the situation where the environment where they are was created by you, by the experimenter. If you would have noticed any tendency to try to transform it somehow by crushing against the limit to make an opening or just adapt it somehow to their own action.
Yeah, I mean, they will certainly chew on anything. So if your environment is made of cardboard, for example, they'll chew it into little pieces. They will try to build nests. If you give them a sort of suitable nesting material, they'll gather it all together. So they do have, and if you give them a substrate that they can burrow into, then they will create burrows. So they do have a little bit of architectural motivation in some senses.
That's interesting.
Trying to stop that type of thing. It's kind of annoying if they're burrowing and digging and chewing, and we want them to just explore.
So perhaps if you would design settings that were made of food components that they could eat, maybe they would try to use the food or eating the food to sculpt something, even if accidentally.
Accidentally, quite possibly. I would be surprised if they have an intrinsic creative motivation other than to create a burrow system. So I think they probably do have an intrinsic motivation to build spaces to live in. That's not been very much explored for technical reasons. It's quite interesting though to think about how that's done.
So now going back to the brain and to the human brain and also the connections, perhaps you could tell us a little bit more about from the biological perspective, also in terms of evolution. Why are there similarities between our own brain and the rat's brain in terms of navigation and how we process navigation?
I think because these elements evolved a long time ago, like probably back in the days of fish, our sort of far away ancestors, the fish that were the ancestors of rats and mice, and also of primates. So we haven't actually really fully explored the fish brain yet, so we don't know whether there are place in grid cells there, but certainly people are starting to find these elements in birds now. So, they're all vertebrates, so we all have this same common ancestor. And I think that the similarities have persisted because these are really fundamental and important functions that have been conserved, is the word for things that persist in evolution. They've been conserved because they provide an adaptive advantage. So, knowing where things are in space is incredibly important for survival. And once you've got a system that works well in the brain, evolution that has just not messed with that very much. Also, it's very tied in with memory. So we think that this system is forming the scaffolding for memory. In other words, it's how the brain is organizing its memories. And again, that's really fundamental and conserved.
And another question before opening the questions also to here in the classroom and also to the audience, I would like to ask you, has to do with neurodiversity, because in your presentation you mention, I think in a more general sense, this navigation system and the orientation systems that our neurons use to locate themselves and navigate through space. And so my question will be, for example, I'm left-handed and I have always had every now and then difficulties with navigation. I don't know if you're familiar with that. And I know that, for example, people with dyslexia also have a tendency to have trouble with navigation, specifically in urban contexts where we have a lot of presence of this kind of grid system, left, right, and so on, to create this kind of orientation. So do you think that these are difficulties we have because of the way the environment is built, or do you think that there's also a correlation with neurodiversity and maybe non-neurotypical individuals would be more propense to have more difficulties navigating the majority of the environments we have? So what could you perhaps tell us about that?
Yeah, so there's quite a range of navigation abilities in the general population. I don't think it necessarily maps to the normal neurodiversity profiles that we talk about, like autism or dyslexia or handedness or anything like that. Like it seems to sort of cut across that, if you like. A lot of otherwise neurotypical people have really quite poor navigation, sometimes to the extent of being extremely anxious whenever they have to navigate somewhere unfamiliar. And we're just starting to try and understand what's different about their brains that makes it difficult for them to navigate. And there's probably many different causes, because we know that there's a lot of different functions go into building up a spatial map and then using it. So for example, some people might have difficulty with this function of recognizing landmarks and knowing how that then tells you which way you're facing. Some people might have difficulty just remembering sequences and so on. Some people have difficulty with mental rotation. So when you're thinking about navigating somewhere where you aren't actually at the moment, you have to kind of do that in your mental imagination. And that requires an ability to manipulate sort of images spatially in your mind, and maybe some people are not so good at that and so on. So there is a wide variety of individual differences. And I think in future, what we want understand is what are the different categories, and then how do we design environments to enable the people with those different deficits, if you like, to still be able to navigate well.
Thank you. Would there be a question here in the room?
Yes, we noted 2 questions.
Could you come?
Oh, yeah.
We have here a meeting owl that will facilitate our conversation.
Great.
So one of the questions I have is, where can't you transfer this knowledge you gained about rats to the brains to the humans? So where are humans different from rats?
Yeah, good question. So we have slightly different senses, for example. So rats are very olfactory. They use their noses very, very intensively, and their vision is very poor. So when rats are navigating, they probably rely a lot more on their sense of smell than we do. And some of that may be just following odor trails. So not even using a spatial map, but just following the path that another rat took and building up a picture of what the various paths are through the environment, which we just can't even imagine because we don't do that. We ourselves are very visual. And so we rely very heavily on being able to kind of see. And if we're deprived of vision, like in the fog or in the dark or something, we can get quite sort of disoriented. We probably have a better ability to do mental rotation. So to be able to, imagine ourselves in another place, trying to navigate to somewhere different. So we're probably able to put ourselves out of the here and now a lot more than rats can. And of course, we have language, which probably helps enable the development of that capability, because with language, you're taking your spatial knowledge and you're trying to turn it into a form that can transfer that knowledge to someone else's brain. So that requires a lot of ability to decode language and also to build up a mental picture when you receive language. All of that's probably something that rats and mice can't do. So there is, we do have a lot of capabilities that they don't have, but that's all built on a foundation that's ultimately the same. So.
Thank you. And the other question I have is about scale. So these principles, do they work the same in a room as they work in a city? Or is there a difference between?
Yeah, that's a really good question. I've been thinking about that a lot. So we do know that there is quite a range of scales that grid cells, for example, can be sensitive to. So I showed you examples where the firing fields, the patches of activity were like 30 centimeters apart. But if you go down to the very, very deep part of the brain, then those distances can be many meters. So that suggests perhaps that the brain is at least trying to make maps on those scales. When we get to kilometers, it's less clear how the brain is mapping that space. And the only person I know who's really tried to address this is Nakhon Molinovsky, whose group in Israel has been studying bats. So they've been studying the way that the bats will fly like tens of kilometers from the home to a foraging patch and back. And so they've been trying to record while they're doing that. And I think we don't quite fully know what the answer is yet, but it's looking like indeed there are place cells that have enormous fields, like will fire over very large ranges. Having said that, I think it's also quite likely that our map of large-scale spaces is kind of mosaic, so it's made of lots of fragments of smaller spaces. So we don't really have one big map of the city, or at least we have a sort of hierarchical map of the city, where we've got very high resolution maps of the little bits of it, like my room and my house and so on, and a very coarse representation of this district and that district. But yes, that's still very much under investigation.
Very interesting. Thank you very much.
Who also would like to make a question? No? Okay. So I think there's a question here in the audience. Milton, would you like to go ahead?
Sure. Hi. Hello, Kate. That was a terrific talk. As always, I'm always learning something new, and and I always get new questions from it. One of them is has to do with Rudolf Arnheim. I'm not sure. I don't know if you're familiar with Arnheim's work, but he wrote a book called The Dynamics of Architectural Space, and he had, in some ways, there's a lot of subtleties in there about how you feel when you're in a space relative to the objects around you. He is a psychologist, I believe, and he's written a number of books that deal with this. This one is from 1977, I think. So this does not coordinate directly with neuroscience research, but I think it's relevant to it, and it'd be worth, I'd like to have some more conversation about it. But I think Arnheim is a source that architects look to, and I think embed some things that are relevant. When I was looking for it on the shelf, it was right next to this one.
Oh, thank you. I'll take a look at that, because actually it may well be relevant to neuroscience. So one type of cell that I didn't mention that's been recently discovered are cells that become active when the animal is at a particular distance and direction from objects in the environment. Mm-hmm. And you can move the objects around and the cell will move around where it fires. And the really interesting thing is that a subset of these cells, once an object's been placed in the environment and the cell has become active, when the object's taken away, the cell remains active in that space. So it's like it's holding on to a memory of where that object had been. Really cool. We don't know what they're for.
Fascinating.
But yeah, but it may be part of building up a much richer representation that has memory and is anchored to objects as well as boundaries. And so that book, I must have a look at that book. It sounds very
sure. And the other one I just wanted to mention is that I've read that the deaf community has a very efficient way of instructing people. For instance, I left my keys in my apartment. Here's where they are. Can you go get them for me? And very efficiently using, I don't know if it's American Sign Language or which one it is, very efficiently being able to direct people how to move through space. And that might also show how the human brain does things when it's more conscious. That has to tie back, of course, to how the human brain does things when it's not conscious. So that might be another source.
Right. So this is like route information, like go through the door, turn left, and so
right.
That's yeah.
I think there's specifically some way of referencing coordinates so that you can say whether it's in the far corner, the left corner, the northeast, the northwest corner, etc. That's what I've read.
That sounds
okay. Thank you, Milton. So we will have to finish soon. Unfortunately, I would very much like to continue this conversation, but we need to pay attention to the time. There's a last question here on the chat from Tatiana. Thank you for this inspiring lecture, Kate. I need to leave for my next meeting. My question, which strategies might you recommend to architects to help reduce spatial unease in their designs? What are some potential applications of this knowledge, specifically in the early design process when architects are engaged in programming and spatial planning?
Yeah, that's a really interesting question. I think for me, the most important thing would be this thing about the sense of direction, because I think a sense of direction, and this is still sort of just a hypothesis at this stage. So we've not really got all the data yet, but my sense is that if you're in a complex space that has multiple subcompartments, you are much less likely to become confused and therefore anxious if you have an understanding of the spatial relations of these compartments to each other in the sort of directional domain. So you have, in other words, the same sense of direction in every room, so that you understand how the rooms are related to each other in their directional relationships. So you need to think, well, how would you make that possible? So if somebody's going out of one room that has opaque walls and going into a room down the corridor that also has opaque walls, how is a person going to maintain a consistent sense of direction across those two rooms? And what can you do to facilitate that? So for example, can you make some of the walls glass, or can you provide some landmark that can be seen from both of the rooms or something like that. So how do you kind of enable the brain to link across these spaces so that it's aligned its head direction cells in all of those different spaces? So I think that's where I would start.
I could imagine now when you were describing specifically an example of a space like in a hospital space, for example, or a dentist practice that one doesn't know very well, and very quickly these would be strategies that would help find one's way in a space that one is unfamiliar with.
Exactly. Yeah.
So thank you very much, Kate, for your time and for sharing your knowledge with us. We have to come to an end now. It was really a great pleasure, as always, to learn from you. So thank you.
Thank you for the invitation. And apologies for the technical fluffing at the beginning.
No problem.
Bye-bye.
Bye-bye.
Bye-bye.